So most of the things that we talked about in the previous set of concepts are going to be reviewed again in greater detail in Bio 182. So I'm going to leave those, especially the concept of speciation and macroevolution. Those are primarily Bio 182 concepts. Microevolution you're going to talk about in detail again in that class. But here we have to know something about evolution. Even looking at the level that we're looking at, which is skin in, as we've talked about before, we must understand evolution because it's impossible to understand a lot of what happens in molecular biology and in genetics without reference to evolution. It's almost impossible to do that, which is why we had to introduce it here. And let me give you an example. This is the first example that we've seen of natural selection in action. Okay, and it is relevant in every HIV patient we've treated, every single one. In fact, it's relevant in essentially every cancer patient we've treated as well, and that's what I work on personally. So the point, though, is this. People think that evolution is saying that things happen randomly, that the chances of things occurring and evolving is just random chance. And that's often been a critique of the concept of evolution, concepts of natural selection. The basis of that critique sounds right, but is flawed. It sounds right because what they're saying is, well, mutation. What's a mutation? Okay, now write this down because there's no slide that says this. A mutation, by definition, is any permanent change in the DNA sequence. The sequence of bases that we talked about, ACGs, we've seen that earlier. If you take those sequences and you change any of those bases, change them at all, we call that a mutation. Now, I'm going to get into detail about what mutations are later in the course, but that's what it is. Okay, now, what can cause mutations? Mutations arise by three primary mechanisms. First, mutations can occur when radiation interacts with the DNA and causes the DNA to change. That radiation could be ultraviolet radiation, could be gamma radiation, any number of things can do that. The second mechanism is chemical compounds that you might be exposed to through your food or through the environment. And the third thing is when the DNA has to replicate itself. Whenever the DNA replicates itself, there's a chance that it's going to introduce a mutation because whenever it makes the copy, it doesn't make the copy perfectly. So those are all the different mechanisms that you can have that can cause mutations. Now, where exactly the mutation arises is random. Exactly what portion of the DNA that gets mutated, what portion of it changes, is random, and when it occurs is random, meaning it can't be predicted, just like we talked about before. Okay. Therefore, their logic says, since that's random, evolution must be random. And the response to that is, no, that's wrong. That's a misconception. Because, yes, it is true that the mutation in any single individual is random, but what happens next, natural selection, is not random. What lives and dies is based on how beneficial it is to that species, or in this case, the virus. All right, so let me show you this. I'm going to show you this in actual patient data right here for HIV. So the example I'm going to use here is HIV, human immunodeficiency virus. That virus arose in the 1980s. That was when it was first recognized. And as with COVID-19, as soon as it came up, people started working like crazy to try to find some way of managing it or curing it or doing something along those lines. And they very rapidly found a compound that actually controlled the virus very well. It's called AZT, or zidovudine. And AZT then was really heralded as the cure. We're going to be able to cure HIV. But something was discovered in every single patient that they treated with AZT monotherapy, meaning they only gave the patients AZT. And that something is illustrated by these two patients. Now, these come from real data. And on this axis, in both of these graphs, you have concentration of AZT. And on this axis, you have a measure of the proportion that are resistant to the drug. Okay, now, first of all, let's talk about the experiment. In this experiment, what they did is this. You take human cells that you're culturing in your laboratory, and those cells are a particular type, they're a type of T cell, that you have, the HIV can infect. And what you do is you take these cultures of cells and you infect them with HIV. So you're not hurting anybody, you're not putting anybody at risk, but you're able to see then how the virus particle is able to grow through that tissue. Now, what you then do is once you've got a good solid HIV population growing in there, you hit it with the drug. And the drug you're going to hit with in this case is zanavudine, AZT. Now, you don't just use one concentration of drug because what you're trying to do is to determine how the virus is responding to the drug. And so what you do is you have a bunch of different dishes and you give them different concentrations, right? So the lowest concentration is 0.001 micromolar, goes all the way up to 10 micromolar. Now notice this scale right here is logarithmic, right? It doesn't go 1, 2, 3 or 0.001, 0.02. It goes up by powers of 10. And when we do this, that's because we're looking at concentrations that are over an extremely big range. All right, now, another thing I want to point out is this concentration up here, 10 micromolar, that's essentially deadly. You can't give a patient that much. You'll kill them. Okay, so what you do then is you take these, you give it the different concentrations, and then you measure the proportion of the viruses that survive. And that's a measure of resistance to treatment. That's what this is, resistance of virions. This is the proportion that survive. Relative viability is the proportion survive relative to the controls. Okay, so in this case then, what this is showing, that dot is showing, is that in the first month, that's what this one is, the first month after diagnosis of this patient, if you take their virus particles out of their blood, grow them in the dish, and give them this concentration right there of the drug, then over 50%, about 60, 65, almost 70% are going to survive. So you give it a higher concentration, a lower percentage survives. That makes sense. More drug, more death, less survival of the viruses. Give it a higher drug concentration, a higher drug concentration, higher drug concentration. What this is telling you is that the higher your concentration that you give the virions, the lower proportion of them survive. But notice, we never got this down to 0. That never goes to 0. Okay, so then the same patient lives for another 10 months. They come back 11 months after they've been diagnosed, and they do the exact same study, the exact same techniques, and this is the data that they get. Now, at this concentration, they didn't even try because 100% survived. At this concentration, where before only about 30% or so survived, now over 70, essentially 80% survive. And now to get the same kind of control, in fact, you never get this control. To get the same kind of control, you got to give them drug concentrations that would kill the patient. All right, then the patient comes back 16 months after diagnosis, and it's even higher now. In order to get any comparable death, you have to essentially give the patient a lethal dose of the drug. Right? So that then is showing that over time, this curve is going out the upper right, which means that over time, resistance is increasing for all concentrations of drug. Okay, that's patient 1. That's actually patient 2. Patient 1, let's take a look at that. In this case, patient 1, after 2 months after diagnosis, got really good control, much better than patient 2 did. But in this case, after 11 months, they come back and now, look, same thing happened. This goes up. And then after 20 months, it's to the point where now no drug concentration is effective except lethal ones. This happens in every patient we've ever treated with AZT monotherapy. In fact, this happens with every patient we've ever treated in any monotherapy with HIV for HIV. This is evolution by natural selection. Obviously not random. If it were random, some patients would become resistant and other patients wouldn't. This is not random. 100% of the patients become resistant. Why? It's mutation. Mutations are still involved. But why is it that they become resistant? Right? That's what we need to understand. So we need to go into the molecules and take a look and see what exactly is happening. What's causing this issue? And the cause has to do with a very, very simple change in the DNA of this HIV. To understand the details of what's going on here, we have to know a little bit about the HIV life cycle. And so this is a picture of the life cycle, sort of a summary that comes from the Center for Disease Control and Prevention. And it goes over more than we're going to go over right now. We'll come back to this and talk about this later. But here's the point. The virus particle is this. It's a very simple thing, and it doesn't have all of the organelles and structures that it needs in order to reproduce itself. In particular, it doesn't have ribosomes. So in order to reproduce itself, what it has to do is it has to infect a cell, and here's the cell, the host cell. And it goes through a series of events, which we'll talk about later in the class, to get its RNA. Now that's what this string is here. This red string right here is RNA. That has to get inside the cell. And when it does that, it gets copied into DNA. And that's what the yellow is here. So the DNA copy then gets integrated into the nucleus, and these are genes now from the HIV itself, and those are going to then be used to produce the proteins that are used to produce the virus particles, and then the cell becomes a virus factory. Okay, that's the very shortest story of what's going on. What we're interested in is right here, right at this step. At this step, there is a particular enzyme that's called reverse transcriptase. Reverse transcriptase comes with the virus, and it gets released into the cytosol of the host cell, the cell that's being infected, and it binds to the RNA strand here, and it reads that RNA strand, and from it builds DNA. Now, we will get into the details of that later, but the point is now, when it's reading that RNA, it has to take DNA nucleotides that are inside the cell here, and those DNA nucleotides include adenine, thymine, guanosine, cytosine, A, C, G, and T, and it has to put those into the proper order based on what it's seeing on the RNA. Okay, and again, we'll get into the details of that later, but the point is it needs those 4 different nucleotides, A, C, Gs, and Ts, in order to make the viral DNA. Okay, now, when it does that, that reverse transcriptase can be sloppy. It isn't always perfect when it makes those copies. That is the critical step here, which we'll talk about here in a moment. But our goal is to block this step. That's what AZT does. Its goal is to block this reverse transcriptase step in copying the viral RNA into viral DNA. What AZT is an analog, something that looks very much like thymine. And so the idea then is this: if we give the patient the drug, then the cell will have thymine in it, but it'll also have the drug AZT in it. The drug, like I said, looks like thymine, except that it has a particular change on it. And that change makes it unable to be used for growing more DNA. So that if you put AZT in place of thymine, then it breaks that whole system. So the reverse transcriptase will be continuing and continuing and continuing, and every time it sees an A in the viral RNA, it knows it's supposed to put a thymine. So sometimes it'll grab a thymine, but every now and again it'll grab one of these AZT molecules instead because it can't tell the difference. When it puts the AZT molecule onto the growing chain, it stops the process right there. If it stops the process, the DNA then cannot be integrated in the nucleus, and therefore we're not going to get an HIV infection. That's the idea. Okay, so here's a sort of cartoon of reverse transcriptase here. And it's got these arms that come out, and here it is bound to thymine. Okay, and so the idea then is when it's bound to thymine and it's making that growing chain, it can't tell the difference between thymine and AZT. So when it does that, it grabs that AZT and will put it in and stop the whole process. Okay. Now here is a space-filling model. It's, I think, a 3D printed model of what the AZT actually looks like. And here's the arm. That's the arm that changes, or actually it's probably here. This is the arm that changes. This is the other arm. Okay, so this is the key. Now, this is normal reverse transcriptase. But like I said, when the reverse transcriptase is making the copy here, it will cause mutations. It will be sloppy. And it will occasionally make a mutation in its own gene. Here's the key. This is a little bit tricky. The gene for this reverse transcriptase, the thing that holds the information about how to make a reverse transcriptase, is on this RNA that's being copied by the reverse transcriptase itself. So when this thing is going to make new reverse transcriptases, it will sometimes introduce a mistake when it was copied in the first place to cause reverse transcriptase to have this little part that sticks out here, and it's shown here in red. And this little part is just sticking out a little bit farther than that, and that's the only difference. But that's the mutation. That's a change in the DNA sequence of the virus, not the RNA sequence, but the DNA sequence, a sequence that's made when it's made into DNA. And that little arm is sufficient for the mutant reverse transcriptase to be able to tell the difference between thymine and AZT. And that means then it will only bind thymine and will not bind AZT. So if this becomes the reverse transcriptase, then this is the one that is going to be resistant to the drug because it can tell the difference between it and thymine. Okay? All right, now that mutation that arises here is random. It occurs randomly. But only in a single individual does it occur randomly. But the point is this. This is only happening in one cell. In a given patient, the patient who's infected with HIV are producing viruses from many, many thousands, millions actually, of cells. And the number of viruses that are produced by a single individual per day is 10 billion. So a single individual who's HIV infected produces 10 billion new virus particles per day. Now, those viruses will all be copied, not all of them, but those that survive will be copied. And when they're copied, they'll introduce mutations. Now, the cell or the virus is not saying, oh, okay, there's AZT, I'm going to mutate here. That doesn't happen. The virus can't think. But some cases it'll mutate someplace, other cases it'll mutate someplace else, some cases it'll mutate someplace else, but there's 10 billion of them. So at some point, you're going to get every possible mutation that can occur somewhere in that population, enormous population of HIV particles. You're going to get a mutation, every possible mutation that can occur. This is one of the possible mutations. The vast majority of these mutations that occur are going to cause the thing to be wiped out. It's going to wipe that particular lineage out. But there's another lineage. There's a bunch of them. Remember, there's 10 billion every day. 10 billion every day. So if one of those 10 billion dies, who cares? If 15 of them die, who cares? If 100,000 of them die, who cares? That's nothing in 10 billion. But all it takes is one that gets this mutation. And that's the point. The point is, it's not random because in the big enough population, every mutation is possible and every mutation will occur in a week in a person's body. So it's not at all, it's random when it's going to occur and exactly which virus particle is going to occur. I can't tell you which of those two things. I can't say it's going to occur at 1:30 p.m. on Tuesday. I can't do that. But I can say over a week it's going to happen in that population of 10 billion per day that are being produced. So that's the key. That's why it's not random. These mutations are going to arise because the population's big. That's why evolution is a concept of populations, not a concept of individual virus particles. Okay, but now what happens if it does? Well, here's the thing. All those mutations are arising in individuals before they're even treated, which means every possible mutation has arisen before we've even started treating this patient, which means this stupid mutation has gotten there already. The good news is this little mutation is only beneficial under treatment. When there's no treatment, this reverse transcriptase, the virus particles carrying this type of reverse transcriptase, are not as competitive as the ones carrying this reverse transcriptase. So the vast majority of patients, when we first start treating, have this reverse transcriptase because this is the best one in a natural human being. Okay, so here we go. So here's the mutations that arise. Remember, every possible mutation will arise. Most of them will kill the lineage off, but it doesn't matter. There's 10 billion every day, blah, blah, blah. Okay, so here's where we start treating. Time is going in this direction to the right. Here's where we start treating. Now notice, this individual has 3 relevant mutations. Now, it's typically more than that. Okay, 3 relevant mutations that change this a little bit. This isn't the only possible mutation that can occur. This is the most resistant one, but there are others where it changes it slightly differently, and that's what this is showing, various mutations that change it slightly, make it slightly more resistant to AZT. The green is slightly resistant. The blue is this one that's fully resistant. Okay, now watch what happens when we treat. When we treat, we kill all the original susceptible ones off, these guys. We kill all those off. We kill off a lot of these that are partially resistant, but not all of them because they're partially resistant. But notice, we don't kill any of the fully resistant ones. So we keep treating, and we keep treating. Well, the more we treat, we get a higher concentration. We finally kill off all those green ones. But notice, no matter how much we give, These guys are the ones that grow back. All right, so now when you first treat, you wipe out most of the viruses. You can see that. But then when it comes back fully and utterly resistant. And now we can give it as much drug as we want and it won't have any effect. That's evolution by natural selection. And that's why it is that we don't use AZT monotherapy to manage patients who have HIV. In fact, what we do is we use 3 different drugs. One that is this type of reverse transcriptase inhibitor, which is what's called a nucleoside analog. We use another one that's a non-nucleoside analog. It binds to this protein and blocks it in a different way. And then we use another one that's a protease inhibitor. So we use this combination of drugs. And what we're trying to do is to get it to such a low population size that it evolves slowly. And not only that, but things that are resistant to AZT are more susceptible to the other drugs. And so we put it into what's called a double bind. We try to get it to a situation where it evolves into something we can treat, which then causes it to evolve back into what we had before. That would be the perfect situation. Okay, so that's the goal here. So we're trying to deal with this issue. Now, this isn't just HIV. This happens in cancer patients too. This is what I study. So when you treat a cancer patient, you kill the cells that are susceptible to the treatment. The ones that survive are the ones that are resistant to the treatment. So over time, in some patients, the treatment fails. And when it fails, the cancer that comes back is fully resistant to the treatment that you've been giving them. So that is a very important issue in medicine. And here's the point. Without understanding natural selection and evolution by natural selection, we are causing problems. Let me give you an example. Ever heard of MRSA? Methicillin-resistant Staphylococcus aureus. It's that flesh-eating bacteria, and the problem is it's methicillin-resistant. It's resistant to antibiotics. Not all antibiotics, but most antibiotics. Why does that happen? We caused it by this mechanism. We caused methicillin-resistant Staphylococcus aureus. Tuberculosis. There are forms of tuberculosis out there right now that are essentially untreatable. If you personally get one of these forms of tuberculosis, we can't treat you any better now today than we could in 1820. And it's because that tuberculosis is what's called MDR. It's multi-drug resistant. It's resistant to essentially all known antibiotics. They arose in Russia, and it's because when we started treating people with these drugs, we did not take natural selection and evolution into account. Because of that, people are dying. We could treat individuals far better, and we do treat individuals far better when we take natural selection into account in our treatments. So that's why it's important. We have to understand evolution. We have to understand natural selection because it's important. If we don't, we will not be able to treat individuals as effectively as we could.